Okay, guys, so I'm back. Video 3 Meiosis. It's a new day, so wardrobe change. Family came home and I got busy and I didn't get to this last night, so I'm doing it this morning. I've also had a location change, so if you're watching the background, now we're in the spare bedroom. So here's what I wanted you to do. I wanted you to see this in all of its long glory. And then what we're going to do, I'm just going to skip ahead here a second. We're going to look at each one of these separately and we'll talk them through. Okay? So this is the big picture. We're going to start out in prophase 1 with a diploid cell. I'm pointing, but that's not doing you any good. So over here, we'll go to metaphase 1, anaphase 1, telophase 1. And then between telophase 1 and with telophase and cytokinesis, we do not have an interphase. We do not have another S phase. We do not undergo any DNA replication. We go into prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis, and then we're going to end up with 4 haploid daughter cells. So that's what we're doing in meiosis. Okay, meiosis 1 has 4 stages. There they are. Here's what they look like. So in prophase 1, we've come through interphase. Interphase is over here. We've come through interphase. We've replicated that DNA. So now every chromosome is made up of a pair of sister chromatids and they're attached to the centromere. And in prophase 1, the centrosomes are moving towards the opposite poles. So there's 2 centrosomes there. See them? They're in yellow. Okay, they're forming their spindle apparati. Okay, each chromosome is made up of sister chromatids. We've got homologous chromosomes coming close together. So the chromosome 1 with chromosome 1, chromosome 2 with 2, 3 with 3. Okay, we're seeing the nuclear envelope fragment like we do. And then there's another term here that we'll talk about, but it's called chiasmata. It's right here. Chiasmata. Chiasmata are going to be points between homologous chromosomes where they're going to come in close contact with one another. It's going to be how they line up. Okay? It's going to be partly what we're going to talk about is we're going to talk about something called crossing over, where we're going to see gene segments from, let me pull it up here. We're going to see gene segments that are going on the non-sister chromatids that are going to exchange DNA. So a little bit of DNA from the dad chromosome is going to exchange over onto the opposite side with a little bit of DNA at the mom chromosome. That was why it was so important for the loci to be the same. Excuse me, that was why we had to have, I think I used the gene for moons on your fingernails, right? It has to be in the same spot so that what you're doing is you're flip-flopping dad's gene DNA with mom's gene for the same trait. Otherwise, you wouldn't be swapping same traits. You'd be trading an apple for an orange, and then somebody's going to have too many apples and somebody's going to have too many oranges. Okay? And where crossing over occurs, you get these little X-shaped regions. That's the chiasmata. So any place they come, the non-sister chromatids are going to come in close contact and they're going to join and they're going to cross over. They're going to exchange DNA. That's called a chiasmata. Okay. Metaphase 1, they're doing just like anything does in metaphase. They're going to line up here on the metaphase plate, the equator of the cell, however you want to call it. The centromeres are going to have their kinetochore and their non-kinetochore microtubules coming off. So you're going to have the kinetochore microtubules connect at the centromere. Oops. Hi. And the non-kinetochore microtubules are not going to connect to anybody. Anaphase 1, we're going to start pulling apart these chromosomes. Okay? The sister chromatids stay attached to one another. It's just the homologous pairs are going to re-separate. But what's happened is we've had the opportunity for crossing over. Okay, so we've had a chance for exchanging DNA. And then in telophase, and so they separate out, and then in telophase 1, and then cytokinesis. In telophase 1, we're going to see all the stuff we see in telophase. We're going to see the nuclear envelope reform. We're going to see the nucleolus reappear. We're going to see all the things that we've seen and talked about in telophase. in mitosis. The difference being is that we've had this genetic material exchange. So I guess really the point, and I don't think I've ever said this before, but when we talk about animal cell and meiosis 1, I think really the point is we're going to get to cross over genetic information. Okay? So what I've got here is I've got all of the words of the things we were talking about. So here's prophase 1. Synapsis, the homologous chromosomes come and they kind of hang out close to one another, and their arms, their tails kind of tangle together. We have crossing over where we exchange the DNA. The X-shaped points where they cross are called chiasmata. Metaphase 1, the tetrads are going to line up. They're a tetrad because there's 4 copies, right? I have 2 sister chromatids from dads and 2 sister chromatids from mom. We call that a tetrad. Microtubules are going to attach. Looks like this. Anaphase 1, all the stuff we talked about, they're going to start to move towards the opposite poles as the kinetochores have attached to the microtubules, and the microtubules are going to start to chain in. Okay. Telophase 1 and cytokinesis. Okay, so in animal cells, just like we said in mitosis, we get a cleavage furrow that forms. In plants, we'd have a cell plate. So remember to keep those separate on your exam, cell plates versus cleavage furrows. Because we're in meiosis, what we're talking about here is no chromosome replication. I think I've said this 3 or 4 times now. No chromosome replication between telophase 1 and prophase 2. Okay, so the end of meiosis 1 to beginning of meiosis 2, no DNA replication. We've still got that tetrad. And then, I went the wrong way, sorry guys. Okay, then we move into meiosis 2. So we're going to have prophase 2, metaphase 2, anaphase 2, telophase, and cytokinesis. And then the thing to remember is that this meiosis 2, looks a lot like mitosis. It's just that we've had that opportunity to exchange DNA back in meiosis 1. So prophase 2, metaphase, anaphase, telophase. So prophase 2, we're going to do what we've always done. The centrosomes are going to migrate toward opposite poles. The spindle apparati, or the spindle fibers if you prefer, are going to form. The nuclear envelope is going to start to disintegrate. The nucleolus disappears. But remember, we're doing this in 2 cells, basically simultaneously. Okay, because remember where those 2 cells came from. They came from right here, the end of meiosis 1. Okay. Metaphase 2, we're going to see that the chromosomes line up. Now, I don't have homologous pairs lined up this time. I just have plain old chromosomes lined up. Okay. Just like that across 2 cells. The kinetochore microtubules are going to have attached at the centromeres. Keep trying to use those same words over so you guys grab that vocab. Okay, at the centromeres. Notice that you've got this exchanged DNA. You've got some red on your blue and your blue on your red. Anaphase are going to separate. So we're in anaphase 2. The sister chromatids separate. In anaphase 1, the homolog separated, the homologous chromosome separated. In anaphase 2, the sister chromatids separate. And then telophase and cytokinesis, all the stages that we've already talked about. Nuclear envelope reforming, nucleolus reforms, DNA starts to unwind and uncoil, goes back into that chromatin state, and we get 2 daughter cells here that have half of the genetic material of the cell that they started with. And it looks, in the words, like this. Okay. Prophase 2, metaphase 2, they look like this. Anaphase 2, sister chromatids are separating. So that's part of how you know. If we talk about sister chromatids separating, then we're talking either about anaphase 2 or about, anaphase in mitosis. Okay? We are not talking about anaphase in meiosis 1. We're talking either about anaphase in mitosis with a T or anaphase in meiosis 2. So anaphase 2 and meiosis 2 is when sister chromatids separate. Okay? And then telophase 2 in cytokinesis. Opposite poles, nuclei reform, the nucleolus reforms, the chromosomes go from that tight wound up condensed, they loosen back up and we get that spaghetti look. Okay? Cytokinesis, same thing as it's always done. It's going to separate the cytoplasm. Nothing new there. Nothing new at all. At the end of meiosis, we've got those 4 daughter cells. They each have half of the DNA of the cell that they started from. But here's the other thing. They're now all genetically different. They're not the same. We've had that crossing over occur. We only have half of the complement. And now what's going to happen is at fertilization, we're going to take one of these and it's going to meet up with an egg or a sperm, depending upon what this is going to end up being, right? Because it doesn't matter. Going to end up meeting up with another complement who's done the same thing. This is how we see traits skip a generation. This is how we see traits that your parents don't have, but your grandfather or your grandmother did, or reasons why reasons why you'll have one redheaded kid in a family because mom and or dad were carrying that trait, but it didn't express in them, right? But they passed on the gene and now it's expressing in their offspring. Okay. Just some comparisons. You know I love the compare and contrast. So these are all things that I've been saying to you as we go through. But they're written because I wanted you to be able to see them in writing. This is one of my favorite ways to reinforce what I've learned, and I've got it separated out into 2. I kind of separated it and blew it up for you. But I love this comparison. I love to be able to look at the difference between these 2 and say, okay, here we go. This is what I've got. Okay, and that really, really, really helps me with the cementing down. Mitosis versus meiosis. Here's the words. We know I love the compare and contrast. We know I love those questions in class. If you're in my in-person class, which you guys aren't, but in the in-person class, well, some of you are, but my hybriders aren't. In the in-person class, I love to put these up on the whiteboard. When does this happen? When doesn't this happen? That kind of stuff. And I kind of didn't do that in this chapter just because the material was heavy, but we'll do some of those in class if you're watching this in advance. Okay, 3 events that are unique to meiosis. Okay, things that happen in meiosis that don't happen in mitosis. Okay, all of them happen in meiosis 1. And these are them. So we get in prophase 1, we get synapsis and crossing over. That was where we get the formation of the chiasmata and we get the exchange of DNA between the non-sister chromatids of the homologous chromosomes. Okay. At the metaphase plate, they line up as tetrads. So instead of lining up like this and separating the sister chromatids, they line up like this and then they separate the homologous chromosomes. And then the third thing is that in anaphase 1, it's what I just said, the homologous chromosomes are separating instead of the sister chromatids. This is what allows those sister chromatids to stay together in meiosis 1. There's a special little protein complex called a cohesin. I know that looks a lot like cohesion, but there's no O in it. It's a special, I know. There's a special little protein complex called cohesin, and that's what allows them to stay together. And then in mitosis, those are what gets cleaved at the end of metaphase. Sorry for that, that keeps popping up. In meiosis, the cohesins are cleaved along with the chromosome arbs in anaphase 1, so they separate at that point. I never ask that question, but it's in case you've got the curious brain going on. Mutations happen when we have a change in an organism's DNA. So we have a change different than the original intent or the original code. Okay? Mutations are going to create different versions of genes. So they're going to create I can't think of a good one right off the top of my head. Different versions of a gene, and we call those different versions our alleles. So there's a vocab word that we're going to use a lot when we get to Mendel, but a different version of a gene is an allele, and it doesn't have to be a mutation. I don't want to leave you with that impression. So um, freckles, no freckles. Those are two alleles of one gene. Okay. Moons on your fingernails, no moons on your fingernails. That's a version of an allele. Do I have the type A antigen on my red blood cells? Do I have the type B antigen on my red blood cells, or do I have none of the above on my red blood cells? And in that case, we're talking about three alleles. We have three variants that we could have. Well, technically four. A, B, none, which is type O, and then AB. Okay? So those are all alleles. An allele is very simply just a different version of a gene. And then what happens is, because of this, we reshuffle those alleles. Remember, we're crossing over, we're exchanging them, we're separating them randomly, and that's all stuff we're going to talk about when we get to Mendel. I've got three mechanisms that are going to contribute to genetic variation. I'm going to ask you probably these on this chapter and probably these again in Mendel, because this is really important that you understand how we get such diversity when we're dealing with organisms that are so similar. And it's the independent assortment of chromosomes, crossing over, and random fertilization. Okay? So independent assortment of chromosomes. When we're forming those gametes, right? So we had that homologous pair and we have dad and mom, right? Well, nothing says that dads always line up on the left and moms always line up on the right. This isn't grade school, boys in one line, girls in the other. It doesn't work like that. They just come up there kind of randomly. And however they do, they do. And remember, I could have this, and now I'm going to swap different DNA between these non-homologous, non-sister chromatids. So there's that. That's the crossing over. And then random fertilization. Which egg is going to meet which sperm? Okay? Or basically that. I don't have an or. Which egg is going to meet which sperm? Okay? And then this is me just talking through those with you. You guys know I like to put it in writing. So independent assortment of chromosomes, this is what we're talking about. So when we talk about possible combinations, okay, we're talking about how many different ways can chromosomes assort into the gametes, okay? When I have 2n is where I start and n is the haploid number. And when we talk about humans, with n being 23, there are roughly 2 to the 23rd possible combinations. That means we're talking about 8 million different combinations. So it's amazing when siblings look alike at all. I mean, short of identical twins, it's amazing that siblings look alike at all when you're talking about 8 million possible combinations of chromosomes that are out there. This is just the visual to go with that, so I'm starting you off here. Possibility 1, possibility 2. I'm only at metaphase. I'm going to get these daughter cells, right? So I have all these different combinations. So 1, 2, 3, and 4. But this is just on, this is just on a 2 chromosome individual. Now let's work that through when we're talking about 23 chromosomes. There's where the 8 million comes in. Okay, what is crossing over specifically? I know I've said it, but there's a solid definition. So crossing over is when we exchange DNA between non-sister chromatids within a homologous pair. What it does is it produces something called recombinant chromosomes. Recombinant chromosomes combine genes inherited from each parent. So normally I would have just had this set of genes that I inherited from mom and this set of genes that I inherited for dad. And if they separated, well, then this egg got just the stuff from dad and this egg got just the stuff from mom. And what recombinant chromosomes do, or does, I think it's do, are going to exchange that DNA, right? So now over here I've got some dad, some mom. And over here I've got some mom with a little dad. And so that's part of the beauty of crossing over. It happens very early in prophase 1. It's when we have those homologous pairs lining up. Okay, remember it's always crossing over is always between non-sister chromatids because sister chromatids are identical. So there's virtually nothing good or not bad either, but there's no advantage to crossing over between sister chromatids. Okay. Again, there's your view, reinforcing your chiasma or chiasmata. Either word is acceptable. Okay, reinforcing that notion. Typically, these chiasmata, these crossing over points, happen further from the centromere. They happen out on the ends of the tails far more frequently than they happen right near the centromere. They can happen close to the centromere, they just don't typically. So the further out on the gene they are, the more likely they are to cross over. Here's our recombinant chromosomes down here. Random fertilization is just like we said. Any sperm can randomly fuse with any egg. It's purely luck. That makes it sound like you are pure luck. Kind of cool, but you are. How you got here is just luck. Even if you or your offspring were in vitro or conceived in not a normal's a bad word, but in not the usual way, right? That's still random luck. Short of, which we can do, short of us profiling the egg and the sperm and choosing very specific genetic traits. And like I said, we can do that. We can design our babies now. We're talking about luck, okay? So the fusion of two gametes, each with 8.4 million possible chromosome combinations, produces a zygote with any of about 70 trillion possible diploid combinations. So, you know, wow. Just wow. Like I said, it's kind of amazing that we look alike, our siblings at all. All right, so what's the evolutionary significance? All semester we've been coming back. If you look back and maybe, and sometimes it's subtle and sometimes it's not, but all semester we've been coming back and talking about natural selection and evolution and what does this mean? Well, generic variation within a population is huge, and it happens because of this variation in offspring and sexual reproduction, but it's what allows in part for natural selection. So what natural selection does is it concentrates or accumulates specific alleles or specific genetic variants that are favored for a given environment. Okay? So if you take pale me and you stick me in a point in time where clothes are not popular, and you stick me near the equator, okay? My paleness, while novel and potentially attractive within a certain tribe during that time, is not going to be favorable for that environment near the equator. I'm going to burn. I'm going to be more likely to develop melanoma. I'm going to be more likely to die young. Because I don't blend in with the environment. I don't have certain key traits and features that are necessary to survive in that environment. It's why polar bears are white and live in the Arctic, and why black bears and brown bears are black and brown and live in the forest. They're all bears, but we have these variants that allow them to survive in those areas. While you could see an albino black bear in the forest, and while the novelty of that black bear might be pretty cool, it's not necessarily going to be favorable for survival in that environment. And that's what Darwin talked about from the very beginning, was survival of the fittest. And he didn't mean strongest, he meant best suited to an environment. Okay? These are just extras that I pulled together in case you want to print them off and you want a full-sized version, or you need that quick reminder. This is my reminder that crossing over can happen, right? Can't remember what this reminder was. Oh, no? I don't remember what that reminder was. Sorry. And then here's just some labeled, pulling in some of those terms: chiasma, chiasmata, sister chromatids with cohesion. Sister chromatids are going to be the same color. Don't forget they're bound. The sister chromatids are held together here at the centromere. Kinetochore microtubules versus non-kinetochore microtubules. Just stuff to try and help you remember. Okay, I think that's it. This one went long. Sorry, 25 minutes. But I'll get it up. So this is the last of meiosis, video 3. Bye!